On to the accessory digestive organs. These include salivary glands, the gallbladder, the pancreas, and the liver. And no, you do not have to memorize 200 known functions of the liver. So know the location of your salivary glands, gallbladder, pancreas, and liver. So here are some of the functions of the liver: making and secreting bile, storing glycogen and lipids, maintaining normal concentrations of glucose, amino acids, and fatty acids in the bloodstream, making and converting nutrient types, such as converting carbs to lipids, making and releasing cholesterol bound to transport proteins, inactivating toxins, storing iron, storing fat-soluble vitamins, making plasma proteins, making clotting factors, phagocytizing damaged red blood cells by Kupffer cells, storing blood, absorbing and breaking down circulating hormones and immunoglobulins, absorbing and inactivating lipid-soluble drugs. There are 3 pairs of salivary glands. They secrete saliva into the oral cavity through ducts. And if you lift up your tongue and stand in front of the mirror and get something that makes you salivate, such as maybe sour candy, you can watch the saliva squirt out of your ducts. The salivary reflex is stimulated by receptors monitored by cranial nerve number 5, taste buds innervated by cranial nerves 7, 9, and 10. Parasympathetic stimulation increases salivary secretion. So your 3 salivary glands, your parotid in front of the ears, sublingual under the tongue, and submandibular under the mandible. The sublingual produces a mucus secretion that acts as a buffer and lubricant. It provides 5% of total saliva secretions. The submandibular secretes buffers, mucins that will form mucus, and salivary amylase, which will break down starches. The cells transport IgA antibodies into the saliva, providing protection against pathogens, and make most of the salivary secretions, about 70%. The parotid glands make about 25%, and they make a lot of salivary amylase. The cells of the salivary glands and ducts. The ducts assist in the secretion of buffers and antibodies. Mucus cells secrete mucins when combined with water, create mucus, and buffers to neutralize. Serous cells secrete the salivary amylase to digest carbohydrates, and lysozyme, an antibacterial enzyme. Saliva is a mixture of glandular secretions. We make about 1 to 1.5 liters of saliva each day. Most of that is water. It constantly flushes the oral surface. The buffers keep the mouth near 7.0 neutral to prevent buildup of acids produced by bacteria. Most of our foods are acidic. Bacteria create acids, and they will destroy our teeth. Saliva contains antibodies, IgA, and lysozyme to help control oral bacterial populations. Well, despite this, we have condominiums of bacteria in our teeth. And saliva mixes with food to form a bolus that can be easily pushed back to the back of the throat and swallowed. The liver has a capsule. It's covered by a layer of visceral peritoneum, and there are 4 lobes: the right, the left, the caudate, and the quadrate. The right lobe is separated from the left lobe by the round ligament here. Well, actually the falciform ligament that then leads into the round ligament. And if you flip up the liver, this is the posterior surface. It's not the way we normally flip up the liver when we're looking at the torso. But you'll see the quadrate lobe and the caudate lobe on the underside. Here you can see the porta hepatis, the hepatic portal vein, the hepatic artery, and the common bile duct. Associated with the liver is the gallbladder that stores bile. The common bile duct carries bile from the liver and the gallbladder to the duodenum. The porta hepatis is the doorway to the liver, containing the blood vessels and the common bile duct. And there's the porta hepatis. The liver is subdivided into functional units called lobules, and the liver contains about 100,000 liver lobules, each about 1 millimeter in diameter. Lobules are separated from each other by an interlobular septum. They're hexagonal in shape, and they're surrounded by 6 portal areas. That hexagon has 6 corners, so there are 6 portal areas. Look for the interlobular septum, the bile duct, the branch of the hepatic portal vein, and the hepatic artery. And you can see the hexagon shape with the portal, the triad here at each corner of that lobule. The portal area is also referred to as the portal triad. It contains 3 structures: a branch of the hepatic portal vein. We know that hepatic portal system is bringing in the nutrient-rich blood from the abdominal viscera, a branch of the hepatic artery proper, and the bile duct. Branches from the arteries and veins of each portal area deliver blood to the liver or hepatic sinusoids of adjacent liver lobules. Liver cells are called hepatocytes, and they're kind of like spokes of the wheel. Like they say irregular plates, but it looks like sheets of hepatocytes, although the sheets are only 1 cell thick. Exposed surfaces are covered with short microvilli. The plates of the hepatocytes are separated by liver sinusoids, delicate blood vessels that are like large fenestrated capillaries. So you can look for the hepatocytes. They're beige here, and they're radiating out like the spokes of a wheel. And and the sinusoids. Blood enters the liver sinusoids from branches of the hepatic portal vein and hepatic artery proper. About 1/3 of blood supply is arterial. The remainder comes from the hepatic portal vein. Hepatocytes next to the sinusoids regulate the solute and nutrient levels by absorption and secretion. In the sinusoids will be Kupffer cells, which are phagocytes. Iron is also stored, some lipids and heavy metals such as tin and mercury that are absorbed by the digestive tract. Sinusoids all drain into a central vein. The central vein of all lobules merge to form the hepatic veins, and these hepatic veins will empty into the inferior vena cava. Hepatocytes secrete bile, and the bile canaliculi, and these merge to form the bile ductules, which then carry to the bile duct in the nearest portal area. Bile plays a role in the digestion of fats, and that role is it emulsifies fats. That is, it breaks larger pieces of fat mechanically into smaller pieces of fat so the enzymes can get around that molecule and digest it. So here are the bile ductules. Here the Kupffer cells are pink. Your sinusoids here are all draining into the central vein here. liver diseases and conditions can lead to degenerative changes in the liver. For example, viral hepatitis and alcoholism. When blood flow from a clot or damaged tissue is constricted, it causes portal hypertension. So there'd be an increased pressure in the hepatic portal system. This will distend small peripheral veins and capillaries. Some of these vessels can rupture, leading to death. And it also forces fluid into the peritoneal cavity, producing ascites. The gallbladder is hollow, pear-shaped on the posterior surface of the liver's right lobe. It stores and concentrates bile. The bile salts break liquid droplets apart, and this is called emulsification. Basically, it increases the surface area for the lipases, or the fat-digesting enzymes, to get in there. There are right and left hepatic ducts collecting bile from the liver bile ducts. Not so important, however, the hepatic ducts unite to form the common hepatic duct. And bile flows from the common hepatic duct into the common bile duct. The cystic duct takes the bile to the gallbladder for storage, while the common bile duct takes the bile to the duodenum. The common bile duct penetrates the duodenal wall and meets the pancreatic duct at the duodenal ampulla. That's probably the most important part about that slide there. Well, the hepatopancreatic sphincter prevents the flow of bile into the duodenum except at mealtime. So here is your duodenum, and here is where all of the bile is coming down here to this area. The pancreatic duct is coming to this area, and here is a close-up of the ampulla. Here's the intestinal lumen, the pancreas, the common bile duct. So this is a critical area in the duodenum. The liver produces approximately a liter of bile per day. Bile flows into the gallbladder for storage. When CCK, cholecystokinin, by the duodenum is released, it causes the bile to enter the duodenum. So the liver is secreting bile continuously, and the bile is going to go up and be stored in the gallbladder. The release of CCK triggers ejection of the bile into the duodenum, where they will emulsify fats. The pancreas is about 6 inches long, divided into pancreatic globules. You can see dead center is the pancreatic duct. The pancreatic duct delivers the enzymatic secretions to the duodenum. Pancreatic juice is a combination of water, ions, enzymes, and buffers. About a liter of pancreatic juice is produced each day. The pancreatic tissue that creates the enzymes is the pancreatic acini. And remember, the pancreatic islets produce the insulin. The major pancreatic enzymes include amylase, to break down carbs, lipase, lipids, nucleases, break down RNA and DNA. Proteolytic means protein digesting enzymes. And these are active. These are secreted inactively. They're activated in the duodenum, and these include trypsin, chymotrypsin, carboxypeptidates, and elastase.